Okay, rock and roll.
All right, let's say we've got all of these processes I'm about to mention are inside the mitochondrial eukaryote cells. In prokaryotes, bacteria and archaeans, they're going to occur either in the cytoplasm or on their outer plasma membrane, their outer cell membrane, depending. Okay, and the process we're going to be looking at first is the formation of that critical molecule, acetyl coenzyme A, or acetyl CoA for short, because that molecule is the fuel to power what's known as Krebs Cycle. And afterwards, we do what's called oxidative metabolism, or oxidative phosphorylation is a common term for that. That's what the book uses. I'll just simplify it a little bit. And of course, that's where oxygen gets used in our metabolism. And that's where you get the big ATP payoff, by using oxygen. Okay, well, those are the three processes that we're going to look at. Now, so, first of all, let's take a look at acetyl CoA formation. Okay, that is going to occur in the matrix of the mitochondria. The matrix, of course, is that fluid-filled inner part of the mitochondria. That's when you get through the outer and inner membranes. And that's also where you have the DNA and ribosomes and stuff like that. Just about every kind of molecule we use for energy purposes is ultimately going to be used to produce acetyl CoA. Okay? Now, the big thing, sugars. Sugars get broken down by glycolysis and into pyruvate acid. So if you start with sugars, we transport the pyruvate acid into, which is a 3-carbon molecule, into the matrix of the mitochondria. There, a gigantic enzyme complex, it's called Pyruvate Dehydrogenase, but this enzyme complex does a process very similar to fermentation. Now, in fermentation, what we did is we took the pyruvic acid, tore a carbon off, released it as a carbon dioxide, and we had some kind of 2-carbon molecule. That was often ethanol or acetic acid. Lactic acid is produced by some organisms and in our own muscles. Even acetone, some organisms will have acetone as their 2-carbon product. In this case, this process is very similar to fermentation, but we're going to do a big difference here. We're going to tear a carbon off, releasing it as carbon dioxide. We have a 2-carbon molecule left. Now, if we just release that molecule free, that would be acetic acid, vinegar. It's a common fermentation process. But what we're going to do is we're going to take this coenzyme A, or CoA for short. We're going to, and the minute we tear that carbon off, we're going to take that 2-carbon fragment and covalently attach it to coenzyme A. Now, acetic acid, when you attach it to something, we call that an acetyl group. It's a 2-carbon group, and it's organic acid at the end, so it's negatively charged in solution. So, now we take this, we have formed acetyl, so we can spell right, acetyl coenzyme A, or acetyl CoA for short. So this is very much like fermentation, except we're not going to release acetic acid free. We're going to couple it to coenzyme A and then further break it down to Krebs Cycle. Okay, so that's acetyl CoA produced from sugars. But also in the matrix of the mitochondria, we can metabolize fatty acids from lipids, triglycerides, in such a way. So we take fatty acids, which are long-chain hydrocarbons with their organic acid at the end, and enzymes cut the fatty acid into 2-carbon fragments and play around with it, turning it into acetyl groups that are coupled to coenzyme A. So we break it in little pieces, and now we generate more acetyl CoA. Now, by the way, that's why fats have more calories, more metabolic energy than sugars or polysaccharides like starch do. Because gram for gram, fats contain more than twice the calories of sugars or polysaccharides. So if you eat a spoonful of sugar, you get so many calories. If you eat a spoonful of fat or corn oil or something like that, you get twice as many calories. May not taste quite as good. And the reason why is this. The fatty acids, you can make an awful lot of acetyl CoA from each fatty acid because you're talking 12, 16, 20, even 24 carbons long. That generates a lot of acetyl CoA. So fats are more energy dense. They have more calories than sugars and polysaccharides because they make more acetyl CoA. That's where the calories are coming from. Now, also, if you use amino acids for energy, most of the time when you eat protein, what's going to happen is most of those amino acids are just going to be used as building blocks to make your kinds of proteins. But if you're on a diet that's high in protein and very low in everything else, you'll actually have no choice but to use amino acids for energy as well. A number of different pathways depending on the amino acid, but ultimately, once again, we generate more acetyl CoA. So, you know, the old saying, all roads lead to Rome, in these terms, all metabolic roads for energy lead to acetyl CoA. Now, an interesting thing about that enzyme that turns pyruvic acid into acetyl CoA, all right, that enzyme called pyruvate dehydrogenase is an example of quaternary structure gone mad. That enzyme consists of 60 separate protein subunits of 3 different types. All 60 of them have to be in the exact quaternary structure for the enzyme to work. The enzyme is so gigantic that it's larger than some viruses and almost the size of a ribosome. You can see this thing under an electron microscope quite easily. Okay. All right. So it's a humongous enzyme that does this. Okay, well, now we've made our acetyl CoA. That's going to be the fuel for our next step, what we call Krebs Cycle. So let's take that. Don't forget, I am trying to go on this relatively fast so that we can all get out on time or even before time. I know we all want to get out of this place, right? Yeah, well, I'm not making any promises. But if you have any questions, don't hesitate to ask, okay? All right, so we've got our acetyl CoA made. Now, we're going to go to our next step, which is called Krebs Cycle. And it's named after the German biochemist Hans Krebs, C-Y-C-L-E, okay, who discovered it, mapped it out in the 1920s, got a Nobel Prize for that kind of stuff. Okay, so we got a Krebs cycle. It's also called the citric acid cycle because citric acid is a major component of this. And it's also called the TCA, or tricarboxylic acid, if you're interested, cycle. They're all God, I can't spell today. C-Y-C-L-E, okay. All right, it's called, any of those are interchangeable terms. All right. Now, what Krebs cycle is, the basic idea is this. We're going to take that 2-carbon acetyl group from acetyl CoA, attach it to something in Krebs cycle, a 4-carbon molecule, to generate a 6-carbon molecule. And then we're going to break that 2-carbon acetyl group down completely into carbon dioxide. In other words, we're going to tear 1 carbon off and produce a 5-carbon molecule. And then later on, we're going to tear another carbon off and go back to a 4-carbon molecule. And it's called a cycle because about the 10 steps that make up Krebs cycle, you start out with this 1 4-carbon molecule, and then you end up at the end of it with the same molecule you started out with. So that's why we call it a cycle. So there's about 10 steps in there. Once again, because some enzymes just work faster than others, to control the whole cycle, we're going to use feedback inhibition to slow down the inherently fast enzymes. Krebs cycle occurs once again in the matrix of the mitochondria. And if you're a prokaryote, it just occurs in your cytoplasm. The enzymes are all there. So, make this big cycle. Okay, we start out with a 4-carbon molecule. And if you're interested, it's called oxaloacetic acid, if you're interested in that. No, we're not responsible for that, by the way. You take biochemical. It's a 4-carbon organic acid. What's going to happen is we're going to take acetyl CoA over here. That's the coenzyme A with the acetyl group. An enzyme is going to tear the 2-carbon acetyl group off of the coenzyme A, leaving the coenzyme A without anything attached to it. That's going to be one product. And it takes that 2-carbon acetyl group, transfers it from acetyl CoA, and covalently attaches it to our 4-carbon oxaloacetic acid. So we got 4 carbons plus 2 carbons from the acetyl group. We end up with a 6-carbon product. And that is citric acid. That's why we call this a citric acid cycle. It's the very same citric acid that makes oranges and lemons and grapefruits and stuff sour. Citric acid is widely used in the commercial food industry as a souring ingredient, make things tart. You look at candies, you look at sodas, they're full of citric acid. But citric acid is a very important intermediary in Krebs cycle. Okay, so now we got the 6-carbon organic acid, citric acid. What we're going to do in a series of steps is play around with the structure of the molecule. At a couple points, we're going to tear a carbon off of it, release it as carbon dioxide. So somewhere, say down here, we go to a 5-carbon molecule. And we release a carbon dioxide. We've torn a carbon off. Now, we play around with it some more, and then eventually we tear our 2nd carbon off and go to a 4-carbon molecule, releasing carbon dioxide again. Couple more steps, and finally we regenerate the original 4-carbon oxaloacetic acid. Now, if you're interested, the book's got all, complete chart of this kind of thing. You take biochemistry, you have to memorize the entire cycle and that kind of stuff. We don't do that over here. Okay, so, as a matter of fact, most of the carbon dioxide you breathe out, the carbon dioxide in your exhale, it's about 5%. That's enough to be toxic. So, if you ever did as a kid breathing into a plastic bag repeatedly, eventually you start feeling all kinds of distress and stuff like that. That's from the carbon dioxide. If you ever gone to a cell incubator where they're growing cells in culture, you usually keep the carbon dioxide to about 5% there. You get a whiff of that stuff and you start coughing, it's irritating and things like that. Okay. So we put out a lot of carbon dioxide. Now, carbon dioxide in the atmosphere is only 0.4%, although we're doing our best to increase that number. It's already half again what it was in the pre-industrial era, so it may well double if we don't do anything about it. But anyway, so there's a lot more carbon dioxide in your exhale than there is in your inhale. Okay, most of that is produced in Krebs cycle. You get a little bit too in fermentation and acetyl CoA formation. Okay, well, so we break the acetyl group completely down. Now, why do we bother doing it? The reason we bother doing it is because it's several different steps, and we don't need to know the details about exactly what happens where, but in several different steps, we are going to generate energy-rich molecules. That's where we're get our energetic payoff. At one step, we are going to produce a GTP. Now, GTP, ordinarily we use that for cell regulation, right? Regulating G proteins. But enzymes can tear the 3rd phosphate off of GTP, pass it to ADP, and make it ATP. It turns out GTP hydrolysis is a tiny bit more exergonic than the energy needs of making ATP. So you can do that. So we can say this GTP is roughly the equivalent of an ATP. You can use that to make an ATP, or you can use it for something else if you wish. Okay, now that's one thing. But we're also going to generate, at different points in the cycle, 3 of these nucleotide coenzymes in what we call the reduced or electron-rich form. So we end up actually 4 of them. And I'm going to put them all over here, although they don't really occur all in one step, but I'm going to put them here just for our purpose. We produce one molecule, something called FADH. And then we produce 3 of NADH. These coenzymes, once again, you can take electrons from them and pass them to something else, or you can pass electrons to them. from something else. They're used to donate or receive electrons in oxidation reduction reactions. Now, the cool thing about this is if you tear electrons off of these guys and pass them to something else, in other words, you oxidize them, oxidation reactions are exergonic. And if you, get your left. And if you take electrons from this and pass it to something extremely electronegative, you release a ton of energy. And that is exactly what our next step is going to do. We're going to take electrons from these coenzymes and ultimately pass them to the second most electronegative atom around, oxygen. The energy released from the coenzyme to oxygen is huge. In fact, it's so much that, there's a little debate, the standard thing is that FADH, each one of them, if you use oxidative metabolism, can generate between 1.5 and 2 ATPs. That's nice. Each of the NADHs can produce 3 or possibly 2.5 ATPs. There's a little bit of debate. Standard thing is 2 and 3. The textbook says 1.5 and 2.5. But, okay, so we'll stick with the old convention of 2 or 3. It may not be totally accurate, but to give us a good idea. But the thing is, what it means is each NADH is packed with energy. If we can take electrons from that, pass it to oxygen, that can give us 3 ATP, or maybe close to 3 ATP. That's awesome. That means a lot of ATP. Now think of this. If we do this kind of math here, each time, if you take pyruvic acid from glycolysis, you take glucose, break it into 2 pyruvic acids, you get only 2 ATP out of that in glycolysis. Now, you take the pyruvic acid, make acetyl CoA, 2 of them runs Krebs cycle twice. So let's see how much additional ATP, if we use oxidative metabolism to deal with these coenzymes, we can get by 2 rounds of Krebs cycle. Well, 2 GTP equal 2 ATP. 1 FADH, that's 2 of those. We get 4 ATP per glucose. 3 times 3 times 2. Whoa! Look at that. 24 ATP per glucose. As long as we use oxidative metabolism, we've got to have that because we've got to pass these electrons from here to something else. Okay, now, compare that to lousy 2 ATP you get per glucose from glycolysis. In other words, aerobic metabolism using oxygen generates about 10 times as much ATP as anaerobic processes. That's why anaerobic microorganisms grow very slowly. That's why. Now, you can grow yeast in anaerobic conditions, no oxygen. But boy, their growth rate is real slow compared to how they can grow if there's oxygen around. And that's why with our cells, which are metabolically very active, if you cut off the oxygen supply, the cells peter out, especially your brain cells, the nerve cells in your brain. They run out of ATP in about 30 seconds, and then you go unconscious, and several minutes later you're dead. We cannot survive anaerobically. Our cells are too active. That's true with most multicellular organisms. There's some species of roundworm that can actually run its whole life cycle anaerobically, but that's very rare. Very few can do that. Okay, so clearly, big energetic payoff from Krebs cycle coupled with oxidative metabolism. That's what we're going to look at next.